going to finish up chapter 3 with our respiratory system. All right, so now looking at that movement. So we talked about the gas exchange and the pressures that drive all that. Now, how do we actually transport it in the blood? So hemoglobin is mainly what is transporting oxygen. So a little bit is dissolved in the plasma, but then the majority is carried by the protein hemoglobin. So this is a protein that's made in immature red blood cells and takes up most of that volume. Now we have 4 different types. We have embryonic hemoglobin, fetal, adult, and then what's called hemoglobin S. And that's only in individuals that have sickle cell anemia. So that S is for sickle cell. Embryonic and fetal have a higher concentration of hemoglobin, and it has a higher affinity for oxygen than the adult or the maternal hemoglobin. So it really is attracted to that oxygen to provide lots of oxygen to that developing infant. The adult has 4 different subunits of that protein with iron. So that's why anemia, which is a lack of iron, affects the transport of oxygen. And where the hemoglobin comes from is the presence of that heme group from iron. So one hemoglobin can carry up to 4 oxygens, so each subunit can carry its own oxygen molecule. Carbon dioxide, about 7% dissolved, and we'll come back to this when we talk about acids and bases. It actually produces an acid called carbonic acid. 23% of that is part of hemoglobin, so it has a higher affinity for oxygen, but can and does transport carbon dioxide as well. What's called the Haldane effect. This is whenever hemoglobin binds to carbon dioxide, its affinity for oxygen actually decreases. So whenever it's carrying carbon dioxide, it doesn't pay as much attention to oxygen. So the more carbon dioxide combined, and then vice versa. So again, if we have a lot of carbon dioxide attached, we don't have a lot of oxygen that's happening. So about 70% of that transported is what's called bicarbonate ions that's dissolved either in the cytoplasm of red blood cells or the plasma of the blood. So the majority of it is transported as a molecule called bicarbonate ions. So some's dissolved, some attached to globin of hemoglobin, but the majority of it is transported as bicarbonate. All right, so the carbon dioxide transport, so at the tissue capillaries, what's happening is carbon dioxide goes into the red blood cells, it reacts with water, and it forms what's called bicarbonate. So when hydrogen from water attaches, it forms bicarbonate and a hydrogen ion. Now chloride ions also enter the red blood cells, and carbonate ions leave. This is called the chloride shift. So as chloride ions come in, bicarbonate go out. And that hydrogen ion that comes off of there combines with the hemoglobin, and it lowers lowering that concentration of bicarbonate and hydrogen inside helps to promote the conversion of carbon dioxide to bicarbonate. Now in the pulmonary capillaries, which is when we want carbon dioxide to leave the red blood cells, this results from the formation of additional carbon dioxide from that carbonic acid, that bicarbonate. So the bicarbonate ions are exchanged for chloride. So again, they go in opposite. So chloride's coming in, bicarbonate's going out, and the hydrogen ions get released from that hemoglobin. All right, so if we look at this reaction, so carbon dioxide and water combine to form what's called carbonic acid, and acids donate hydrogen. So when carbonic acid, what happens is it breaks down into a hydrogen ion, which is why it's called an acid, and then this bicarbonate. So essentially the carbon dioxide is being transported and stored as this bicarbonate ion. And an enzyme is used for this called carbonic anhydrase that catalyzes both of these reactions, both carbon dioxide forming carbonic acid and the acid breaking down into the bicarbonate. So that's carbonic anhydrase. So again, at the tissues where we have carbon dioxide levels being higher, then the bicarbonates removed using that bicarbonate chloride antiporter, meaning again they're going in opposite directions. So that bicarbonate diffuses out of the red blood cells while chloride diffuses in through that antiporter. So that exchange maintains neutral electricity because it's one negative ion coming in and one going out. So there's no change in electrical potential. So removing that from inside the red blood cell, though, promotes the transport of that carbon dioxide because more can then come in to form more carbonic acid and then more bicarbonate. But we need to keep the concentration of bicarbonate low, which is why we need the chloride coming in so they get exchanged. Kind of moving through this. So we have the carbon dioxide leaving out of the blood. So again, what's happening here, we have our carbon dioxide combining with our water here. So that carbonic anhydrase catalyzes the formation of carbon dioxide in our bicarbonate. Okay, so we're getting that coming in. Again, it breaks down into carbon dioxide that then again diffuses out, and then our bicarbonate here from the breakdown of carbonic acid. Again, it was formed whenever that bicarbonate and that hydrogen ion combine, but here's this chloride ion or this chloride shift. So the chloride ions are going out, and that allows the bicarbonate to come in to the red blood cell. Again, it forms that carbonic acid, and then it's going to release the carbon dioxide that then goes into those alveoli, and that release of that carbon dioxide allows more bicarbonate to come into the red blood cell, and it continues the process. All right, now the systemic gas exchange. So this is in the tissues. So the pulmonary is going there at the gas exchange at the lungs. The systemic is at the tissue level. So now that carbon dioxide is coming in. Here we have our red blood cell. So again, this is our tissues that are having, so they have all this carbon dioxide buildup from metabolism. Well, that carbon dioxide comes in. It's going to form with water to form that carbonic acid. Then that carbonic acid breaks down into bicarbonate and hydrogen ions. Well, now in order to keep everything moving, we need to get the bicarbonate out of the red blood cell. So that's where chloride comes in. So chloride comes in to exchange that. And now we have oxygen diffusing out of the blood and the carbon dioxide binding to that hemoglobin. All right, now things that affect that transport. So chemoreceptors. So this is going to look for changes in pH. So again, as that carbonic acid is becoming bicarbonate and hydrogen ions, it's going to change the pH. So we have receptors that look for that. We also have receptors that look for changes in that partial pressure of oxygen and carbon dioxide. And these central chemoreceptors are at a place called the chemosensitive area in the brainstem at the medulla oblongata. So think about our brainstem going down, it's the oblong, hence the name part, which is where the respiratory center is located. Then we also have peripheral, so not at the central nervous system, but we have peripheral chemoreceptors that are near the carotid and aortic bodies that are also detecting those changes. So this oxygen hemoglobin dissociation curve describes how much hemoglobin is saturated at different oxygen levels or values. So think about our partial pressure. So at about 104, hemoglobin is 98% saturated. When that pressure decreases, say to about 60, now we're only 90% saturated. And again, and this is due to that affinity of hemoglobin for oxygen as that partial pressure changes. So then if it further goes down to 40, which is what's happening at our tissue level, now that partial pressure, now that oxygen is going to leave and go into the tissue again by diffusion. So now the hemoglobin's down to 75% saturated. So again, by changing the partial pressure of oxygen, it allows oxygen to come off of hemoglobin and move into our tissues. But this is used as a reserve if that partial pressure level decreases even more, say during exercise. Then we have that reserve amount that can be used to provide oxygen during exercise. And again, just understanding the concept behind these are not numbers that you need to know, but just understanding as the partial pressure goes down, the oxygen affinity goes down. So less of it is saturating that hemoglobin. And this is that dissociation curve. So again, it's at the max affinity. Okay, so it's most saturated at about 104 partial pressure of the oxygen in the lungs. As we go down, though, in the partial pressure, which is like in our tissues, now the oxygen is going to dissociate or come off, or down to about 75%. Whenever the partial pressure goes down to about 40, then it can go down even further during exercise, which causes more of the oxygen to come off of the hemoglobin to provide oxygen to those tissues. And now we have another effect, another person's name needs to get added in here. So we have Boyle's law, and we have all the laws in the other recording. Now we have the Bohr effect. And this is the effect of pH on the oxygen dissociation curve. So again, pH is determined by the amount of that bicarbonate. So what happens is the pH of the blood goes down, the amount of oxygen bound to hemoglobin also decreases. And this makes sense because if we have a lot of, so if the pH goes down, that means we have a lot of hydrogen ions, which means there's a lot of carbon dioxide, and we need to have that exchange happening. So we want oxygen to come off of hemoglobin so that carbon dioxide can go on. So again, this is due to that decrease in the pH. Whenever those hydrogen ions combine, it changes the shape, and now oxygen can't bind to the hemoglobin because it's a protein, and pH alters the shape of proteins. So whenever we have a more acidic environment, the change in that protein shape prevents oxygen from binding. And then also whenever carbon dioxide binds, again, that affinity or the binding of oxygen goes down. So if we have a lot of carbon dioxide, then our acids are going to build up because of carbonic acid. That's going to change the shape of hemoglobin. Carbon dioxide attaches, that prevents oxygen from binding as well. All right, and this is just showing the effect on that curve. So again, our red line here is our normal, and again, 40 partial pressure here for oxygen, again about 75% or so is saturated. But what's going to happen whenever our pH goes down or our carbon dioxide goes up, also temperature goes up, now oxygen is going to have less of an affinity to hemoglobin. It goes down from about 80 or so down to about 60 whenever we have high amounts of carbon dioxide or a lot of hydrogen ions and the pH drops. Our main effect of that pH and the partial pressure of carbon dioxide is on, again, how much hemoglobin is attracted to oxygen, or that affinity. So again, the higher that pressure for carbon dioxide causes a larger change or drop in the pH. Again, that drop in the pH increases respiratory rate. And that sensitivity of carbon dioxide is valuable for maintaining that pH. So again, I said we would talk more about bicarbonate whenever we talk about acids and bases in later chapters. Now, temperature can also affect, as we'd seen on that dissociation curve. So an increase in temperature also decreases affinity of oxygen to hemoglobin. So as our metabolism goes up, we need more oxygen. So again, if we're exercising and we're sweating, we want oxygen to come off hemoglobin and move into our tissue. So it's kind of the same thing as whenever that pressure really, really drops low, even below 40. So that affinity of oxygen decreases. Again, our dissociation curve goes over to the right, like we had seen there a little bit earlier. Now, whenever the temperature increases, the rate of ATP, so our energy, is also going up. So again, we're exercising, so we're increasing that ATP production. So more carbon dioxide is entering the blood because, again, we're going through respiration, which increases carbon dioxide, and that gets converted into the bicarbonate and the hydrogen ions. Those hydrogen ions decrease the pH. So that's going to increase the respiratory rate, which is going to help get rid of that excess carbon dioxide. The transport is also affected by a molecule called BPG, which stands for 2,3-bisphosphoglycerate. A lot easier to say BPG. This is a byproduct that's made during the metabolism of glucose. So again, as glucose is being broken down to produce ATP, that's increasing the carbon dioxide production, but it produces this byproduct called BPG. It binds to carbon dioxide to decrease the affinity of oxygen because, again, we need more oxygen for aerobic respiration if we're breaking down glucose. So we need oxygen not to be attached to hemoglobin. We want it coming off into our cells. So again, that affinity goes down and it releases more oxygen than normal. So kind of the potent trigger for that increase is whenever we have low amounts of oxygen in the blood. So again, now our atmospheric pressure is lower at higher altitudes, for example. So that causes the partial pressure of oxygen in the alveoli to change. So in the pulmonary, at the capillaries, it goes down. So now the blood holds less oxygen for delivery to the tissues. So whenever that's happening at these high altitudes, this BPG helps increase oxygen delivery because, again, it's coming off hemoglobin a lot faster by moving that dissociation curve over to the right. Now, it's also affected by, of course, oxygen, but also our carbon dioxide effect. So that BPG enhances that Haldane effect, since the hemoglobin with less oxygen can bind more carbon dioxide. So again, because hemoglobin is losing its oxygen, it means it can attach to more of that excess carbon dioxide that's present due to that increased metabolism. All right, now how do we control our ventilation? All right, so first of all, there's a local control, so not even talking about the brain, but just some local control here that happens. So there's pulmonary capillary perfusion. This is the flow of blood to the alveoli through the capillaries. Okay, then there's also what's called the pulmonary ventilation perfusion coupling, and this is the relationship between the ventilation of the alveoli and to the blood flow. So not 100% of cardiac output is completely saturated with oxygen, and this is due to what's called the physiological shunt. So there's an anatomical shunt. This is due to that deoxygenated blood from the bronchi and the bronchioles mixing with the blood at the pulmonary veins. Then we have the blood that passes through the pulmonary capillaries without becoming completely oxygenated, again, shunted blood. And then we have the physiological shunt, and this is the combination of the anatomical shunt and the blood where it's incompletely oxygenated. And this is about 1 or 2% of that cardiac output. So there's some control of our ventilation coming in at the just within the lungs itself. Now, the ventilation perfusion coupling can be disrupted, though, by that blood flow since they're related to each other. So blood flow coming in can alter it, as well as insufficient airflow at the alveoli. So at the local control, we have blood flow that plays a part, as well as airflow. Those are the two main contributors. So regional distribution of that blood flow is determined mainly by gravity, but also the partial pressure of oxygen in the alveoli. So that low pressure causes the arterioles to constrict. So like in our little arteries coming in, so they constrict, and that shunts the blood flow away from the lungs to where alveoli are better ventilated. So that increases that dead space, but then it allows the blood to go where there is gas exchange happening and not wasting it on alveoli that are not functioning. And then in other tissues, that low partial pressure causes the arterioles to dilate, and that dilation allows more oxygen to actually be able to flow across during that. So in the alveoli, if we have a low partial pressure, they constrict to move the blood elsewhere. If the tissues, those arterioles dilate, it allows more blood flow and more oxygen delivery. Now, the neural control of ventilation is the primary way to regulate that. So it's under voluntary control. So whenever we're eating or speaking, but yet whenever we're sleeping, it's under involuntary control. So there's both a voluntary and an involuntary control of our ventilation. So the respiratory centers there in the brain. So again, the medulla oblongata, that's where our medullary respiratory system is. And what we have there is what's called the ventral respiratory group, or the VRG. This produces kind of that normal involuntary rhythm of our breathing. So there's a collection of neurons there that are activating both inspiration and expiration. We have what's called the Pribotsker complex, which is part of that part of the VGR that establishes kind of the basic rhythm of the pulmonary ventilation. And then we have the dorsal respiratory group, so ventral and dorsal. So the dorsal group receives input from chemoreceptors as well as mechanicoreceptors to modify that respiratory rate. And so again, just kind of the normal rhythm is the ventral respiratory group, looking more at those chemoreceptors, looking at changes in pH or things like that, or mechanoreceptors. That's our dorsal group. We also have the respiratory system and the respiratory center and the pons. So still the pons and medulla both in the brainstem, but we have the pontine respiratory group that's going to regulate that pulmonary ventilation rate. Some of these only help with inspiration, while others only help with expiration. There are some though that do both. But this connects that medullary center, it connects to it, and appears to play a role in switching between inspiration and expiration, so kind of that individual control. Now the chemoreceptors, again, thinking about that dorsal center, so the effects of the partial pressure. So first of all, whenever we have hypoxia, this is a decrease in oxygen level. So it's whenever it's below, so hypo meaning low. So when the oxygen levels drop, what's going to happen is the carotid and the aortic chemoreceptors, again, receptors that are looking for changes, whatever they detect a drop in that partial pressure that's at least 50% of a change, that's increased by stimulation to the respiratory system to keep it active despite the decreasing oxygen levels. So again, it's kind of switching that control to those chemoreceptors whenever that partial pressure drops too low. So that chemoreceptor, so if we think about back to our cranial nerves, so we have the chemoreceptors in the medulla oblongata. So here in the carotid body, we have these chemoreceptors, and using the glossopharyngeal and the vagus nerve, they are going to send that information to the medullary respiratory system in the dorsal area, and that's going to allow, again, this green here, it's stimulating it. So even though the partial pressure drops, it's still going to keep ventilation occurring. Now the carbon dioxide also affects our respiratory rate. So the major regulator of gas exchange is actually through that carbon dioxide at rest or even during exercise. So even a small increase in carbon dioxide triggers large increases in the rate and depth of that gas exchange. So again, main contributor is based on the carbon dioxide. So it takes more than a 50% drop to activate in oxygen to activate it, but small changes in the partial pressure of carbon dioxide have an effect. So two terms here that go along: hypercapnia, that's whenever we have too much carbon dioxide, hyper meaning hyperactive, high, and then hypocapnia would be lower than normal carbon dioxide. So that chemosensitive area and that medulla oblongata is more important for regulating that partial pressure and pH. So again, back to that carbonic acid and the amounts of carbon dioxide present. And then the carotid bodies respond rapidly, though, to changes in the blood pH that indicate too much carbon dioxide due to that intense exercise as we're going through metabolism to provide energy. So that regulation, again, everything's kind of written out here on this side, but again, we have homeostasis. So if the blood carbon dioxide increases, then those receptors in that respiratory system, that's going to detect that increase of carbon dioxide, and that's going to increase our breathing. So we're going to increase ventilation, trying to get rid of that excess carbon dioxide. So again, those effectors, once we increase carbon dioxide, now we're going to bring the pH back to normal, and then our ventilation goes back to normal and breathing is returned. Now, the same thing can happen though the other way. So if we don't have enough carbon dioxide, so if that decreases, again, those chemoreceptors in that respiratory center are going to be detected, and now we're going to decrease the breathing. So we're going to be slowing it down, trying to keep in the carbon dioxide. Once that carbon dioxide level returns, then our breathing returns to normal. So too much carbon dioxide increases our rate of respiration. Too little is going to slow it down. So again, just kind of putting that here. So those chemosensitive areas in the carotid and the aortic measure those changes in pH, which is again indirectly due to the carbon dioxide. Again, carbon dioxide combines with water to form that carbonic acid, and that production of acid lowers the pH. So then they respond directly though to changes in pH. So the lower pH is going to increase the rate because we have too much carbon dioxide, so we need to get rid of it. And then if it goes up too much, then we're going to slow our breathing to bring carbon dioxide back to normal. Now, the mechanoreceptors are what's called the Herring-Brewer reflex and how it affects the respiratory rate. So this reflex limits the depth of our inspiration, so how much we're breathing in, so it prevents overinflation of the lung. So again, thinking about mechanoreceptors, they're thinking about like the movement, the stressing or stretching of something. So we don't want our lungs to overinflate. So these stretch receptors are going to keep an eye on that. And so what happens is they use action potentials to trigger that back to that reflex. So if they stretch, then they have an inhibitory effect on respiration and causes expiration. So again, we're not overinflating those lungs thanks to that Herring-Brewer reflex. Now the central and limbic systems are also involved. So this controls the rate and depth of breathing of both voluntary and involuntary by that cerebral cortex. So the rate changes during exercise. Of course, we need to increase that ventilation to the respiratory system. So the highest level of exercise can be performed without really changing our pH. That's referred to as anaerobic threshold. So that means, again, if an athlete is used to working out, then their body adjusts and they can maintain a normal pH even though they're going through a lot of metabolism and building up a lot of carbon dioxide. So they have what's called an anaerobic threshold. Now beyond that, if the pH drops below that threshold, that's going to stimulate an increase in ventilation, trying to get rid of that excess carbon dioxide. And then we also have apnea. This is kind of a common term we hear about, like especially sleep apnea. And this is the absence of breath. So whenever somebody stops breathing, and that's whether it's voluntary or involuntary, is referred to as apnea. And emotions can also affect our respiratory rate. So again, during crying or hyperventilation, if someone's really excited, emotions, and that's part of the limbic system, also can affect our respiratory rate. And then some other modifications. So even activation of touch, so like thermal and pain receptors. So if we touch something hot, we kind of start breathing a little more heavily or increasing that ventilation. Sneezing and cough reflexes alter ventilation, as well as body temperature can increase ventilation as well. So again, just kind of looking at the major regulatory pathways here, just kind of a summary figure and whether they have stimulating, so increasing respiration or decreasing respiration. So again, we have like our emotions there in the limbic system. We have those chemoreceptors looking for changes in pH and carbon dioxide. Same receptors that are in the carotid and aortic bodies. That Hering-Brewer reflex that prevents overinflation there of the lungs. We also have our muscles and joints that are causing changes to respiration, as well as touch, temperature, and pain receptors. And then finally, just looking at the effects of aging here on the respiratory system. So as we age, our vital capacity and maximum pulmonary ventilation, minute volume, and gas exchange all decrease as our lungs start to age. That residual volume and dead space, so the air left over and those alveoli that don't participate in gas exchange, those increase. The ability to get rid of mucus, so it kind of sits there in the lungs, just being able to cough up and get rid of that mucus and get rid of those microbes gets harder. Compliance, which is how easily we breathe in and out, that increases due to a loss of alveoli. And then gas exchange across the membrane is reduced. So it's just less oxygen coming in and carbon dioxide going out. So all of those are ultimately decreasing respiratory function as we age.